First of all, I would like to thank Alessandro and Paolo for giving me the opportunity to be here today to tell you a little bit about some of our most recent work at the high-throughput imaging facility at NCI NIH. I'll start with a very big disclaimer / apology. I'm not a physicist. I'm a biologist, and so everything that I'm going to tell you today is going to be really geared toward the assay development and the biology more than the technology per se. Having said this, I'll nevertheless start by giving you a very broad and fairly quick overview introduction about high-throughput imaging. And then in the second and third part of the talk, just to really bring home the message that high-throughput imaging, it's an extremely versatile and powerful technique. I'll tell you a little bit about two projects that were recently run and completed at HiTIF in collaboration with Tommy Stelly, which is one of our most fruitful collaborators at NCI. Really at the core of high-throughput imaging, or HTI, there is always an imaging-based assay. And this is a way, of course, using microscopy, to measure the activity of a biological pathway by measuring changes in the localization or levels of a cellular or one or more cellular markers. And the idea is to perform an experimental perturbation of our system, and then through our imaging-based assay, measure a phenotypic change. And this is really not much different of what's going on also in low-throughput experiments in labs throughout the world on a daily basis. What's really unique with high-throughput imaging, and that's the high-throughput part of the high-throughput imaging, is that in this case, what we're really doing is performing not just one or few experiments at the same time, but performing up to hundreds of thousands of experiments per experimental campaign. In addition, as I'll show you in a little while, thanks to automated image analysis, we can really output up to tens of thousands of results for up to tens of thousands of cells per sample. So just by multiplying these two numbers, it's really immediately apparent that the size of our data sets can go up to millions of data points. And finally, high-throughput imaging is also known as high-content imaging or high-content screening, and this is because for each and every cell, we can output up to 1,000 different cellular features if, of course, the assay needs to do so. High-throughput imaging or high-content imaging was originally devised as a technique to screen large collections or libraries of drugs, so in the drug discovery field, but now it's also used to profile compounds, so profile their activity based on the phenotypic effect that they have on cells. In addition, several academic siRNA screening facilities, including here at the ICGB, have used this technique also to screen libraries of siRNAs to systematically study gene functions and dissect molecular pathways. And I'll show you a little example of this in a little while. But then high-throughput imaging, it's also now used more and more, not just to test a large number of experimental treatments to identify few experimental treatments that might have a biological activity, but rather use this technique also to look in a small number of experiments at a large number of cells. And this is going to be particularly interesting and useful for looking at extremely rare but biological important events. From a practical standpoint, high-throughput imaging, it's really a combination of several different techniques. We usually start by seeding cells in multi-well imaging plates, which are usually either 384 or 96 well based in a 96 or 384 well format. The idea is that each well in these multi-well plates is treated with a different perturbing agent, usually either a small molecule, a drug, or siRNA. And then treated cells are further processed and stained entirely according to the cell-based assay in question. And this is the part that usually changes from cell assay to cell assay. Stained plates are then imaged with high-throughput microscopy, and I'll get a little bit more in detail about this part in a little bit. And the images generated by high-throughput microscopy are then analyzed by automated high-content image analysis software to produce a certain number of cellular features that are then used to describe the phenotype of cells in a high-throughput manner. And then these cellular features and the phenotypes are then used to rank or score the cellular the perturbing treatments based on their effect of cells. And so now I'll quickly go through high-throughput imaging, high-throughput microscopy, and the automated image analysis. So what can we actually use to generate a high-throughput imaging assay? The most common markers are stably expressed fluorescent proteins genetically fused to cellular markers of interest. But it's also possible to perform immunofluorescence in a high-throughput format. And so it is possible to detect the localization and or levels of endogenous proteins in a high-throughput format. In addition, our facility and other facilities around the world are now using more and more fluorescent in situ hybridizations to probe at the single cell level and in a large number of samples, either, for example, the position of genes or genomic loci at the single cell levels in hundreds of thousands of cells, or the positions and number of single RNA transcripts. And finally, as demonstrated again by the siRNA screening facility here at ICGB, it is also possible to use a fluorescently labeled substrate for de novo DNA replication or de novo protein synthesis to actually visualize these processes in a high-throughput format. So as far as the high-throughput microscopy, high-throughput imaging, high-throughput microscopy part of the protocol is concerned, again, the idea is to use multi-well plates. And we are equipped with high-throughput microscopes that possess automated stages and autofocus mechanism to image each and every of these wells in a fully automated fashion. Furthermore, the microscopes can be programmed to acquire several images on a per field basis that we usually refer to as fields. And for each and every field, we can actually acquire images, of course, in multiple channels. And then depending on the high-throughput imaging setup, certain high-throughput imaging systems can also acquire three-dimensional information by acquiring Z-stacks. And finally, it is also possible for certain high-throughput imaging setups to actually acquire, to actually run live cell experiments or time-lapse experiments to further acquire information about the temporal dimension of biological processes. It's immediately clear that by multiplying all these different factors, all these different dimensions, the number of images acquired on a per day basis can immediately skyrocket up to about 100,000 images per day. So from a practical standpoint, usually researchers tend to focus either on this part and then maybe probe several thousands different treatments, but maybe limiting or completely eliminating Z-sectioning and time-lapse, or rather using fewer experimental treatments and maybe focusing on getting more detail, more information about the three-dimensional information and or the temporal dimensions of biological processing questions. This is just an example of one high-throughput microscope, and this is the one that we have, the one that I know the best, and this is why I'm showing you this, but of course there's other ones in the market. We're using Perkinelmer Opera NIPCO spinning disk confocal microscope. It's equipped with 4 excitation lasers and 3 cameras for ultra-fast concomitant imaging. And it's also equipped with 4 high numerical aperture water objective. And our workhorse, and most of what I'm going to show you later on, is going to be with a 20x and a 40x objectives. And just as a side note, I didn't mention this, but it's not, we can image multiple plates in a screening campaign, so that a lot of screening facilities are actually equipped with robotic automation equipment to automatically load and unload plates in and out from the instruments to increase the throughput. So I mentioned that the number of images generated on a per day basis can be in the order of hundreds of thousands. So manually analyzing the images, it's unpractical, if not impossible. So what we really need to take advantage of is automated image analysis software, content image analysis software. The idea here is that in a completely automated fashion, in more than 90% of the cases, we use an image of the DAPI channel or Hux channel, so a DNA stain that stains the nucleus, to segment objects and then identify single cells. The nucleus, the primary object, is then used as a seeding object for a secondary compartment, such as the cytoplasm, if a specific cytoplasmic staining or a membrane staining is present, and or other tertiary compartments, such as, for example, vesicles or spots, as I'll show you in a little while. Regardless of the actual image analysis workflow, the result of high-content image analysis is a collection of several numerical features that describe what's present in the image at the single cell level. The advantages over manual analysis of images or visual inspection of images is that it's a high-throughput method, so that we can really process a large number of images in a very short amount of time. It is quantitative and unbiased, and so these largely eliminate user-introduced bias in the quantification of biological processes. And as I mentioned before, it actually outputs data at a single object level, and so we are not limited to averages or medians, like in the case, for example, of normal fluorescent plate readers, which are normally used in the screening process, especially in pharma companies. And finally, this technique and automated image analysis can actually output a large number of cellular features, and so the technique is multiparametric and can be used to analyze complex cellular phenotypes. In terms of what kind of features can be quantified with automated image analysis, the first and most intuitive class of features is counts, so that it is possible to count nuclei or spots per cell, but it is also possible to measure position and distance of objects, of subcellular objects, such as, for example, the relative distance between 2 spots in different colors in the same nucleus, or, for example, the difference between a spot object and a cellular hallmark, such as the nuclear envelope. Changes in intensity at the single cell level, in intensity levels can be measured, but also changes in texture, i.e. in the pattern of fluorescence intensity and or in the morphological distributions of fluorescence intensity inside the cell. In addition, it is also possible to measure changes in size or shape, And finally, the technique also allows the quantification of relational cellular properties, such as, for example, whether a cell is actually a single cell or a cluster as measured by the number of nuclei present in the same cell. Or in addition, for example, we can actually measure the context of a cell based on the number of adjacent cells. In terms of assay formats, this is fairly arbitrary but useful classifications of high-throughput imaging assays in different classes. The first one being the typical large-scale screens, again, such as the one performed in pharma companies for drug screens or the genome-wide siRNA screens. In this case, the number of experimental treatments performed or used is in the order of 100,000. And this means that the number of cells imaged and analyzed per experimental condition is fairly low, in the low hundreds, usually 200 or 300. We and others have adopted a hybrid approach, which I'll refer here to as focus screen. And in this case, the idea is to use selected libraries of reagents, either siRNAs or compounds, where the function of the single RNA or compound is fairly well understood. In the case of siRNAs, for example, this would mean screening libraries of siRNAs that are targeting all genes belonging to the same family, same pathway, or that are all belonging to, or that have all the same enzymatic activity. A typical example, for example, would be a library screening all kinases present in the human genome. This allows screening libraries that are much smaller, in the order of several hundreds to few thousands, but also this allows to acquire more cell per, and image more cells per experimental treatment, and possibly also at a higher magnification. Another extremely fruitful approach, in our case, also in the hands of other labs around the world, has been using high-throughput imaging as a deep imaging technique. And what I mean here is to rather limit the experimental treatments to a few tens, but rather imaging a very large number of cells. And again, as I'll show you in a little while, this is extremely useful to identify and precisely quantify rare events in cellular populations. And finally, a very exciting approach is the use of high-throughput imaging for multiplex experiments, where the number of fluorescent probes, it's in the orders of hundreds or thousands, and the number of experimental treatments is actually in the order of few. But I won't talk about this today. So now let me switch gears and tell you a little bit about the first project. This is a screen that was set up to identify suppressors of the Hutchinson-Gilford Progeria syndrome phenotype. So a little bit of introduction about Hutchinson-Gilford Progeria, or in short, HGPS. HGPS is a premature aging disease, and it's been for a long time that at the molecular level, more than 90% of Progeria cases are actually caused by a recurrent de novo point mutation in the lamin A gene. And the lamin A gene, it's actually one of the critical components of the nuclear lamina, which is a proteinaceous structure underlying the nuclear envelope. And from a functional standpoint, lamin A and the nuclear lamina have been involved in a variety of important nuclear processes, such as, for example, transcription, signaling, DNA repair, and so on and so forth. The point mutation in the lamin A gene actually activates a cryptic splice sites, which leads to an internal mRNA truncation, which leads to the production of an internally truncated protein version of lamin A, which is known as progerin. Progerin acts in a dominant fashion in Progeria patient cells, to cause some striking nuclear defects. The most well-known being the dramatic change in the shape of the nucleus when compared to the shape of nuclei from healthy individuals. In addition, the Mistelli lab and others also showed that at the nuclear level, progerin and progeria patient cells show three other striking hallmarks. The first one being the downregulation of other components of the nuclear envelope. The second being the upregulation of the DNA damage response, possibly indicating that there's increased levels of endogenous DNA damage in these cells. And finally, the downregulations of important heterochromatin markers, such as, for example, trimethylated histone H3K27, and other factors involved in heterochromatin formation. So Nard Kuben, an extremely talented postdoc in the Mistelli lab, actually wondered whether he could set up an assay based on high content imaging in which he could identify the factor or the factors that are actually causing or they're actually involved in this transition between the healthy and the progeria patient state. And even more importantly, he was interested in finding possible factors that, when knocked down, could actually lead to a suppression of this phenotype. So to address this problem, he went on and generated stable cell lines inducibly expressing different versions of the lamin A protein fused to GFP. And he created three cell lines, one expressing the wild-type lamin A protein, one expressing the mutant pathological version of lamin A called progerin, and also a control version of progerin, which was predicted not to have a biological effect. And the idea here was to induce the GFP-tagged version of progerin and then measure the nuclear phenotype using high-content imaging. The first control he did was to actually check for localizations of the GFP-tagged version of lamin A and progerin, and he could actually demonstrate that they localized as predicted, and GFP progerins actually caused nuclear invaginations and defects in a nuclear shape. These induced proteins were expressed at levels that were comparable to the endogenous version of lamin A, and most importantly, at least from a qualitative standpoint, it was immediately evident that the GFP-tagged version of progerin caused the downregulations of nuclear envelope proteins, of heterochromatin factors, and increased levels of endogenous endogenous DNA damage. He then used automated imaging and image analysis to actually, again, segment the nuclei, and then using 4 channels, quantify in the same cells the levels of progerin, the levels of lamin B1, and the fluorescence intensity of phosphorylated histone gamma H2AX spots. And lo and behold, he could express at 4 days post-induction, he could actually see in a quantitative fashion now decreases in nuclear envelope proteins, heterochromatin factors, and increased levels of endogenous DNA damage. He then selected lamin B1 and gamma H2AX because these were the markers that actually gave the best Z prime factors, the largest differences between negative and positive controls. Nard then decided to screen an siRNA library, and the idea here was to basically transfect cells and at the same time activate the productions of GFP progerin, and then look for factors that actually kept the cells in a healthy state. And this is just controls. This is just to show you that, again, he could induce progerin, and induction of progerin caused a decrease in lamin B1 levels, increase in phosphorylated histone in histone gamma H2AX, and positive control technical control on siRNA against GFP, which leads to the silencing of GFP progerin itself, could actually suppress this phenotype. So armed with these controls, it went on and fished for ubiquitin ligases that, when knocked down, could actually show at least a partial reversal of the levels of progerin, levels of lamin B1, and levels of gamma H2AX. One of the most prominent hits in the primary screen that was then validated in secondary siRNA screens to rule out possible off-targets, siRNA off-target effects, was the CANDY1 gene. CANDY1 was particularly interesting because it's actually part of a protein complex that also includes RBX1 and CAL1. And CAL1 and RBX1 also score pretty high during the primary screen, thus indicating that this might actually, CANDY1 might actually be a bona fide modifier of the Progeria phenotype in cultured cells. Nard is very thorough, and so he went back and decided to test CANDY1 knockdown on all the panel of markers, Progeria patient markers, that heterochromatin and nuclear envelope markers that I showed you before, and knockdown of CANDY1 actually rescued all the markers in the inducible cell line. Another very important experiment was to test the effect of CANDY1 knockdown not only in the inducible cell line, but also in the actual cells from Progeria patients. So these are primary fibroblasts. And what he could see was that he could nicely rescue the effects on heterochromatin levels and nuclear envelope levels, but not on the DNA damage levels. But this was pretty much in line with previous observations in our obtained in the Mistelli lab and in other labs. And furthermore, some of you might have noticed that knockdown of CANDY1 actually causes a decrease in the levels of progerin, but the downregulation of progerin per se was not enough to actually lead to the suppressions of the progerin phenotypes, because when progerin per se was induced only at 50% than the quantity used in the screen in the previous experiments, he could not see a partial rescue. So I told you that CANDY1 is a component of a multi-protein complex, and indeed what this complex does, among other things, is to actually ubiquitinate and target for proteasomal destruction the NRF2 gene product. NRF2 is a transcription factor that when senses oxidative stress, increased levels of oxidative stress, it actually translocates to the nucleus and activates the transcription of a battery of antioxidants genes that protect the cells from the effects of oxidative stress. And in fact, when Nard knocked down CANDY1, he could see an increase of NRF2. So now the idea was, is NRF2 involved actually in the Progerian patient phenotype? And the idea here, the hypothesis was, is it actually protecting cells from a progerin? And this seemed to be indeed the case, because when a constitutively active version of NRF2 was transfected in cells expressing GFP progerin, what Nard could see, again, thanks to high-throughput imaging, was a complete reversal of the Progeria patient phenotypes. And I won't go through all the experiments because there were quite a few of them, but Nard hypothesized that progerin might actually affect NRF2, and he was actually able to demonstrate it, and that NRF2 impairment of NRF2 activity actually led to increased levels of oxidative stress and protein damage. And so Nard, thanks to high-throughput imaging, was actually able to demonstrate that NRF2 exerts a protective role in cells, and that progerin blocks NRF2, thus leading to Progeria patient defects. So just to summarize this first part, high-content imaging can be used to systematically screen gene function to identify pathologically relevant cellular phenotypes. So now let me move to the second story. In this case, we decided to use high-throughput imaging not for screening, but rather to detect rare biological events among large populations of cells. So chromosomal translocations are caused by DNA breakage and repair of chromosomal fragments originating from different chromosomes. And they are one of the leading causes of cancer, especially in hematological cancers. And in fact, the genetic consequences of chromosomal translocations are, for example, the fusions of two genes that lead to the production of a gene product that is oncogenic. Alternatively, chromosomal translocations can lead to the translocations of a very active promoter in front of a gene that is not normally transcribed and leading to the expression of improper expression of these genes in tissues and that where it shouldn't be expressed, thus leading again to cancers. The golden standards to quantify, to detect these events, these translocations, is metaphase spreads and break-apart FISH. And the idea here is to have a green probe and a red probe designed to hybridize towards two regions of the same gene. If the gene is broken and it's translocated, the two probes should localize on two different chromosomes. And this is what people observe all the time. There's two main drawbacks to this technique. The first one is that metaphase spreads are extremely labor intensive, and also the visual analysis of where the probes are, whether they're close or apart from each other, it's usually user driven. So in order to test whether it was possible to actually scale up and to make this process more quantitative, Bharat Borman, again from the Mistelli lab, decided to test whether he could adapt break-apart FISH in a high-throughput format in interphase nuclei. And the idea here, again, is to design two probes in green and red here. These are DNA FISH probes against the same chromosomal region. These probes are adjacent. And design a third probe against a putative translocation partner on a different chromosome. The idea is that in case these chromosomal fragment, this gene is intact, green and red should be close together, and the blue probe should be somewhere else. In case of a DNA breakage and separation of the two chromosomal arms, the green and red probe should be now separated. And in case of translocation of these chromosomal region to this chromosomal region, we should now see a concomitant separation of green and red and a concomitant proximity event of blue and red together. So to test this hypothesis, Bharat decided to choose a model system of anaplastic large cell lymphoma. In this kind of tumor, there is one of the recurrent causes of this tumor is a translocation between the ALK gene and the NPM1 gene. And so what Bharat did was to design two complementary FISH probe sets that labeled either the ALK gene or the NPM1 gene with green and red probes, and then use matching far red probes indicated here in blue to look for translocations. So the workflow here again is to generate the probes for the two different genes, treat cells and stain them in FISH with 3 colors plus DAPI, and then use high-throughput imaging to acquire tens of thousands of cells per experimental condition. And then, with high-content image analysis, automated image analysis, what we did was to segment the nuclei based on DAPI and then find the spots in all 3 colors and then calculate all the distances between spots and calculate minimum distances between spots in different color to measure for proximity or separation. And then use single allele statistical data analysis to quantify these events. This is just a stitched image of 35 fields at 4dx. Each field has 6 slices. All the images are zip projected. So it's immediately clear, and this is just one experimental condition. So it's immediately clear that we can acquire a very large number of cells, typically more than 10,000 cells per experimental condition. Now, having so many cells and so many images, again, as I told you before, we had to use automated image analysis. This is just an example of how the segmentation works. We have the segmented nucleus. The software detects spots in all 3 channels, and then we can obtain merged images where we can see whether the green and red spots are close together and where the blue spots are. And we have not just single cell resolution, but we actually have single allele resolution. So this is an actual experiment. What Bharat did was to get a negative control cell line, MAC2A cells. These are ALCL cells that are known not to carry a translocation. So this is our negative control. And as you can see here in this inset, almost all MAC2A cells actually had the ALK gene intact. So green and red probes were together and the blue was somewhere else. Bharat also took a positive control cell line. Again, this is an ALCL cell line that is known to carry a translocation. This was previously published, so this is known. And as expected, what we could detect using this image analysis workflow was that indeed green and red were separated and blue was now together with red. And this happened not on all alleles, but at least in one allele for most cells. And finally, in order to test whether we could detect small changes in the frequencies of these breaks and translocations, we treated MAC2A cells, our control negative cell line, with a high dose of ionizing radiations that is known to cause DNA damage, thus hoping to detect increases in breakage and translocations. And this is just the complementary probe set that shows the same thing. So I showed you the images, I showed you the segmentation. Now the task is to actually collect all this data and actually analyze it. So I told you the image analysis workflow. The image analysis produces data sets at the cell level and also at the single distance level for each and every cell, for each every red spot, for each and every green spot, for each and every blue spot. In order to detect ternary events, so whether a green spot, whether a red spot was really close to a blue spot and at the same time far away to the green spot, we really had to index all the objects all the way down to the spot level. We collected all the image analysis results and created extremely large single object data tables and fuse all the data tables, filter them based on the number of spots per cell, and then we use statistical analysis software for plotting and generate statistical summaries. So what do these data look like? Just a little bit of quality control on the fish detection efficiency. What you can see here is that as previously published and as expected, our fish probe sets correctly determined that the vast majority of MAC2A cells actually carry 3 copies of the ALK gene and 2 copies of the MPM1 gene. Whereas the K299 cells, and again, as previously described by others, are actually composed of mixed subpopulations of cells carrying 2, 3, and 4 alleles. But the really interesting information came out, and the very interesting result came out from the analysis of the distributions of minimum red-green distances on a per red spot basis, and also on the analysis of red for red distances. Here, what you can see is that in 99.99% of the cases in the control cells, green and red spots are 4 pixels or closer, which represents a distance of about 1.2 micrometers, according to our imaging conditions. As expected and nicely, the K299 cells actually revealed that there is a population of about 45% of the red spots that are actually far away from the green spot. And this is what we expect, because these are the cells that carry the translocation on only one of the 2 alleles. And this is nice because you can actually see that we can actually see the 2 subpopulations of alleles in the same cell that are either translocated or non-translocated. And this was also true when looking at red for red distances, because you could see that in K299 cells there is a subpopulations of alleles that it's not only close to where the red spot is not only close to the blue spot, but it's actually further away from the green spot. We could actually visualize all these events at the single allele level, and as one would expect, treating cells with ionizing radiation also led to an increase in the separation events, as indicated here, that once quantified at a single cell level actually showed statistically significant differences between the ionizing radiation treated versus untreated MAC2A cells. So our technique is extremely sensitive, and it's capable of detecting changes of rare events. And most importantly, this was also true of translocation events, which have a frequency that it's even lower than breakage events. It's anywhere between 0.07% all the way down to 0.4%. Nevertheless, again, even if these percentages were very small, since we are imaging so many cells and detecting so many translocation events, the confidence intervals on these estimates are pretty tight, and so that we can actually detect statistically significant differences. This was just as a proof of principle, but Bharat used the same technique to actually address fairly relevant biological question, and this is just an example of what he did, was to use the same technique in a medium throughput fashion. Here we're not talking about thousands of samples, but tens of samples, to actually demonstrate that chromatin structure around these translocation sites, ALK1 and NPM1, actually affects DNA breakage frequencies and translocation frequencies. So in summary, today I showed you 2 possible uses of high-throughput imaging. The first one being the use of high-throughput imaging for screening biological treatments to identify treatments, either siRNAs or compounds that actually have a biological effect. And the second one being instead an application in which we are not screening a large number of samples, but rather use high-throughput imaging to detect rare and quantify rare biological events. And I started with a disclaimer. I'll finish with a disclaimer. This is what I showed you today is not these are not the only possible applications of high-throughput imaging, and what I showed you is very much filtered through the lens of my experience and of the assays that we are running at a high-throughput imaging facility. There's a lot of other applications that this technique is good for, including screening of primary cells, 3D cultures, and so on and so forth. And I didn't really have the time to go go through this today. But there's excellent sources of information online about everything high-throughput imaging. So I refer you to these sources, and I'll be happy to tell you more at the end of the talk if you want to know more. And now I really want to thank the people that actually did the work, Nard and Bharat, which of course are supervised by Tom Mistelli, and Ty Voss, the former head of the high-throughput imaging facility, which was heavily involved with the first two stories that I just showed you. And with this, I thank you for your attention, and I'm open for questions.